Okay, Chapter 18, Section 2, Nitrogen Excretion and the Urea Cycle. Now keep in mind, the urea cycle is our body's mechanism for taking that free ammonia that's been released due to amino acid catabolism by the mitochondria, getting it out of the mitochondria into the cytoplasm into the bloodstream so that the kidneys can get it out of the body. So what we're going to be looking at in this section are these steps right here in this urea cycle. How to get it out of the mitochondria, and then how to go through and remove those amino groups and make the urea that will then be transported by the blood and out of the body. So, first thing, during the catabolic reactions of some of the amino acids to get at the carbon skeleton, which can then be further processed into, different steps of the citric acid cycle, acetyl-CoA, whatever. Got to get rid of that amino functional group. So it's released as free ammonia in the mitochondria. Well, you can't our cells do not have the mechanism for transporting the ammonia across the mitochondrial membrane out of the cell into the bloodstream, and doing so in such a way that it's not going to be damaging to the rest of the body. So we need to package it up, move it around from one organelle out of this mitochondria to the cytoplasm, and then repackage it, molecularly speaking, into something that's a little bit more tolerable. So, first thing, take this inorganic ammonia and attach it to an organic compound. Specifically, we're going to take carbonate. We're going to attach the ammonia to the carbonate, and in the process of doing so, we are going to take a little bit of energy from ATP. So now we take, carbonate, CO3H, add ammonia, add an inorganic phosphate. So, what have we done? C12 phosphate, the amino functional group. Okay, we can do this. This is something we can work with now. So you take free ammonia, carbonate, you end with carbamyl phosphate. Now, in the process of doing this, the carbamyl phosphate synthetase. Okay, synthetase means it's an enzyme that synthesizes something. What's it synthesizing? Carbamyl phosphate. What it does is it's taking ATP, stressing this bond between the oxygen and the phosphate so that you can get this nucleophilic attack from the oxygen here on the bicarbonate. There's that third oxygen I didn't count when I was counting on the last slide because it's now more associated with this inorganic phosphate group. Releasing ADP, and you now have an intermediate carbonic phosphoric acid anhydride. Well, our amino group, that nitrogen is now going to nucleophilic attack there, electrophilic attack, so we're swapping bonds here. Nitrogen to the carbon, that carbon-oxygen bond's now going to go to the phosphate group, because remember, it's going to be 2 single bonds, 2 double bonds. So that phosphate's released, gave us the energy to bring in this nitrogen. Now you have the carbon-nitrogen here at carbamate. Well, we can't do much with the carbamate. So we're going to take the energy from a second ATP, and we're going to add that terminal phosphate right there to that carbon. So now we have NCOB and 2 H's. First ATP to bring in the nitrogen, the second ATP, well, it's going to power this next step here. carbamyl phosphate is going to be added to ornithine. This nice, what is it? 3-carbon, 2-amino functional group molecule. Ornithine transcarbamylase. Transcarbamylase. So you're taking carbamyl and you're transferring it to ornithine, producing citrulline. Carbamyl phosphate can't pass through the mitochondrial membranes. Citrulline, on the other hand, can. Now, I can't remember if it's a direct diffusion across or if there's a transporter. I think there's a transporter involved, but don't quote me on that. So the citrulline goes from the interior of the mitochondria, ends up in the cytoplasm. Cool. Still can't do anything with the citrulline. Citrulline has 2 nitrogens in it, but notice it's a nitrogen, carbon, nitrogen, the rest of the carbon skeleton. So, still can't do anything with those nitrogens. Get rid of those amino functional groups. So what do? Well, first we got to add some energy back in. Got to recharge everything here to power the next couple of steps. But instead of just taking the terminal phosphate off of an ATP, we're actually going to take the ATP. We're going to cut off the 2 terminal inorganic phosphate. So the beta and the gamma, phosphate 2, phosphate 3, are cut off. And then, what is this? Arginosuccinate synthetase synthesizes, and here's what it synthesizes, arginosuccinate. Well, this cytoplasmic enzyme cuts off, as I said, the 2 terminal phosphate groups off of ATP, and then adds AMP, this nucleotide, to that carbon that was from carbamyl, which was from the carbonate. Why do this? Well, it powers the addition of, and you can't see it here behind my big head, so look on the handouts for this lecture, that it powers the addition of aspartate to our citrulline here. So what you end up with is what is referred to as arginosuccinate. Arginosuccinate is a 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 carbon skeleton with some inserted nitrogens and an amino and a carboxyl functional groups on the side. So what we're looking at here, ATP, adenosine, 1, 2, 3 triphosphate. The 2 terminal phosphate groups cleaved off, and this carbon right here with the carbonyl. This second bond here is now going to nucleophilic attack on that phosphate. So now you have what is referred to as citrial AMP. The citrial AMP is an intermediate, which the arginosuccinate synthetase is going to take this and the aspartate and rearrange the carbon oxygen AMP here. It's going to rearrange that carbon oxygen bond to the carbon now nitrogen, as you see here. So you have 2 carbon skeletons with this nitrogen bridge connecting them, hence arginosuccinate. Aspartate added in here, as you see, gives you this arginosuccinate, which is going to cleave right here. This is the leaving group, which is fumarate. Fumarate, if you remember from previous chapter, can then be transformed into malate or any of the amino acids, or turned into a fatty acid. Whatever is going to be happening. The remaining of it, the arginine, as you see right here, has 2 amino functional groups and 1 carbon. And then all of this, which that carbon, 2 amino groups of arginine, gets released by arginase. There you go. Carbon, carbonyl, 2 amino functional groups, that's urea. The remaining carbon skeleton is ornithine, which can be taken back up into the mitochondria to take another carbamyl and start this process all over again. So when you look at it, some of the words I've been using, some of the intermediates, sound familiar because they were previous 3 chapters. Aspartate, oxaloacetate from the citric acid cycle. Hey, body has too much ATP right now, so you don't need to go through. It's missing some amino acids. Okay, well, turn oxaloacetate into aspartate. Since oxaloacetate can't leave the mitochondria, but aspartate can, out here it gets turned back into oxaloacetate, or it becomes arganosuccinate, which becomes fumarate, which can then, recombine and do other things, become malate, which can be taken up by the mitochondria. It's one of the steps. And, so here's some different cycles that we talked about in the previous 2 chapters. Well, it turns out this arganosuccinate, well, this is a crossroads. It can either go this way or it can go to the urea cycle that we just talked about. Arganosuccinate becomes arginine, cuts off the terminal carbon with the 2 aminos. You got urea, which now is the ornithine, which goes back in. Carbamyl phosphate, get your citrulline back out. And the citrulline, which can become arganosuccinate, which can then this way over to this group, or this way into this cycle. So a lot of things we've talked about, a lot of the students many times get lost. Okay, this one, which cycle is it talking about? Which shuttle? Which all of the above? Yeah, that that ammonia you need to get rid of that becomes carbamyl phosphate, which becomes citrulline. The citrulline outside the cytoplasm becomes arganosuccinate, but you know what? It doesn't have to complete this. It can go this way. Or that aspartate that was, the oxaloacetate becomes aspartate, which can be converted into arganosuccinate, which can then go this way and become ornithine. These different things will feed into each other and out of each other in so many different ways, depending upon what other signals are coming from the rest of the cytoplasm, all those metabolic activities, high levels of this, low levels of that, remember the threshold things, can push one way or the other, signals it's getting from other cells in the tissue, or signals it's getting from those hormones we talked about in the last section of chapter 18 here, can push one way or the other. So as I said, there's lots of crosstalk between these. Lots of overlap. Transporters, malate, alpha-ketoglutarate transporter, glutamate, aspartate transporter. Which one is it transporting across at which given time and which direction depends on all the other signals going around in the cytoplasm at that time, at that given instant. As I pointed out, the aspartate arganosuccinate shunt. Okay, the aspartate, is it going to go this way or is it going to go that way? If it goes this way, it's going to become fumarate or alpha-ketoglutarate or something else in the cytoplasm. But if it goes this way, it's going to become arganosuccinate, which means you're going to get going to be making releasing urea and then making of the ornithine, which is going to come back in and blah 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 blah. 2 completely different metabolic pathways that just happen to intersect at a couple of steps. And then when it's time to split, which way do those intermediates go? Just because it was coming from this one cycle Doesn't mean it's always going to have to go this one way. Hey, look, this is a carbon skeleton. That's all this is. It's a carbon skeleton. Is the carbon skeleton going to be reformed, become a fatty acid, become part of a pyruvate, to become gluconeogenesis, to become this, to become that, or is it going to go the other way? So that the cell can get rid of that buildup of ammonia in the mitochondria, turn it into urea, which can then be excreted out and cleaned out of the system. Threshold levels of different things are going to be pushing one way or the other. Because remember, the aspartate's going to have, has a nitrogen on it, so it's going to have to get rid of. Okay, we're going to go this way. Get rid of, because we got to build up. Or I have too much amino acids now we need to get rid of. We're going to start shunting things over to making fatty acids or gluconeogenesis. So we've got to get rid of the nitrogens. All right, we don't have enough of the amino acid pools. We don't have enough. We have too much fatty acids already. So we need to go this way over that way. How do we control? Well, first way, do we have the enzymes? Seriously. I mean, every cell in your body has the blueprint on how to make these enzymes for this, okay? It's just, are those genes readily available? As I said at the last section of the first lecture, not all the genes are going to be readily available at any given time, depending upon which cell it is. So can they get access to it? And how quickly can those genes be transcribed and then translated? So you get a functioning polypeptide or a functioning protein to do either of these cycles that we've just talked about. And then again, you know what? Right here's a great control point. Carbamyl phosphate, the first one leading off. If this one's shut down, well, the whole urea cycle gets shut down because nothing's feeding into it. It's all stuck. If this one's go, 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 guess what? You're making stuff that's going out of the mitochondria and the cytoplasm, and away that cycle goes. So you can regulate this, allosterically regulate it. So it's not on off, but it's just high levels of on, low levels of on. Go, 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 kind of go, So if the cell has high levels of acetyl-CoA and high levels of glutamate, high levels of acetyl-CoA means, hey, you know what? We've got lots of sugars that just went through the glycosidic pathway. We've made lots of pyruvate. Or we've had lots of fatty acids, lots of beta oxidation that we talked about in chapter 17. So we've got lots of acetyl-CoA, so cell's happy there. And we've got lots of glutamate, which means we've got lots of amino acids. So we don't need to keep all these excess amino acids we got going on here. What can we do? Well, acetyl-CoA glutamate in high levels of arginine, amino acid, so you got lots of amino acid, your pools are high. You could take acetyl-CoA glutamate, and the enzyme N-acetyl-glutamate synthetase is going to form N-acetyl-glutamate. Now, N-acetyl-glutamate on its own, but it turns out this is an activator, allosteric activator of carbamyl phosphate synthetase 1. Remember, carbamyl phosphate synthetase 1 takes carbonate, ammonia, and a phosphate group and makes carbamyl phosphate, which will then combine with ornithate and make citrulline. And everything goes on. Turns out, on paper, this reaction should be extremely pricey, energy-wise, extremely endergonic. But, as I pointed out in weird ways on multiple other slides in just this lecture, many of the steps that we've been talking about here piggyback the steps of other cycles, other metabolic pathways. So this urea, synthesizing urea to get rid of the ammonia, for all intents and purposes, is actually quite cheap, if not almost free, because the other metabolic processes are paying the toll. They're paying the toll. This is just coming along for the ride. It's piggybacking. Many of the metabolic or enzymatic reactions that are just for this urea cycle and urea cycle only, relatively cheap. The cost is paid for by the expensive ones of other metabolic pathways that are extremely exergonic to begin with. Now, it turns out that the catabolism of amino acids, you get a lot of this ammonia. Well, what do? Turns out, well, the ammonia can be very detrimental. So as we see, you take the ammonia, you turn it to urea, out with the waste. But there's a problem with that for some individuals. They have a defect in one or more of the enzymes in this process. So they get a buildup of the ammonia, buildup of some of the intermediates. They can never actually get rid of the ammonia, can never get rid of the urea because they're not making urea or not enough. I was originally thought, well, just do a protein-free diet. Can't. They were just saying, well, stay away from animal proteins. You know what? That's the whole basis of vegetarian and vegan diets. And I know there are vegetarians and vegans going, oh, but you can do this. They're so much different. The amino acids, the proteins, and those are different. No. Yeah, the amino acid sequence, the primary and secondary, may be different, giving you different tertiary, quaternary, but it's still the same amino acids. I'm not going into, they're more readily available, any of that. No, it's still the same amino acids. You still have the same problem of the catabolism, the breakdown of the individual amino acids, so you can get free carbon skeletons to gluconeogenesis or fatty acid, anabolic, buildup, whatever. So what do? Well, here's two examples. You use aromatic rings, these benzoate rings. These benzene rings, these organic compounds. So you have benzoate and you have phenylbutyrate. Benzoate combines with glycine, phenylbutyrate combines with glutamine, 2 amino acids. What I mean they combine is you see right here. Here's our benzene ring. Here is our amino acid glycine. It binds here, and if I remember correctly from my organic chemistry, it will cleave right there, giving you this 2-carbon chain, which can then become acetyl-CoA. Off it goes. And that amino will stay right here attached to the benzene ring, taken out with the trash. This right here, the glutamine, also will combine, replace this coenzyme A right here. That amino will remain with the benzene ring, the carbon chain, off to the races, be used elsewhere.